Greetings. The content in this instructional video is a brief detailing of the history of L.A.B.S and how it came to be used in our current L.A.B.S testing trials. If you haven't watched your occupational tutorial video, we recommend you do that now. Now, let's continue. It began earlier this year, 2001, when the RBC amoeba was discovered by our Westmost Science Department. It was a late night at the facility when a microbiotic re-engineering project finally bore fruit. The facility was observing various amoeba cultures when one began to spiral out of control. Scientist Dale Weatherford-Mesh was the first to discover the culture in a state of absolute disarray. While in the previous hour the culture had been perfectly fine, it was now overflowing with amoebas reproducing at a frantic rate. Dale took the liberty of harvesting a sample for further study, while he sent the overgrown culture to be destroyed. He immediately took to the microscope in hopes of discovering the issue, which is when he found that none of the amoebas in the sample possessed the same structure as those originally placed in the culture. Dale had made a startling discovery. Somewhere along the amoeba's perpetual reproductive cycle, one must have mutated, and began consuming and destroying every other amoeba in the culture. This revelation was groundbreaking, and the news was quickly sent to our main office, which then ordered domestic tests to be performed immediately upon the new breed of amoeba, in order to discover the inner workings of this strange, violent microorganism. Their first test involved simple observation of the reproductive methods under certain stimuli. They tried many different organic materials, but only one made the amoebas go haywire: red blood cells. The consumption of these organic materials prompts the amoebas to abandon their original method of reproduction. Instead of splitting into two daughter cells, the amoeba explodes into many pieces, each reforming to become their own daughter cell, which isn't something that anybody had seen in all of their years at the department. This is how the department gave the amoeba its official scientific name, the erythrocyte eukaryote. From there, it was observed how the amoebas seemed to cooperate with each other, sticking close to each other, resembling a single mass. They would all move in the same directions, stretch branches out to collect organic matter to feed. It seemed as if these microorganisms were a marvel of teamwork. It was then decided that the name for this collection of amoebas should be psyllium. From there, a request for the green lighting of the substance in the main facility's testing trials was sent out, and soon accepted. Now you, the facility's newly hired converter, will be taking part in the psyllium trials. This concludes Volume 1. Thank you for listening. If you desire to listen to the other volumes of psyllium documentation, please contact our L.A.B.S main facility record keeper for a copy of the VHS. Keep in mind that these volumes are still in production,